Hello everybody, my name is Faustine Ravon and I'm currently doing a PhD in the Faculty of Pharmacy at the Université Libre de Bruxelles in Belgium. Today I have the chance to present a part of my research project which is about in vitro and in vivo evaluations of the tolerance of a new and innovative anti-tuberculosis drug combination by inhalation. Let's start with some background to explain how this project is born. Tuberculosis is a mycobacterial infection caused by the pathogen Mycobacterium tuberculosis. This disease represents one of the top 10 most common causes of death in the world. Tuberculosis is spread from person to person through the air, affecting principally the lung. Even if it is preventable and curable, 1.5 million people died from tuberculosis in 2020. Current treatments use at least 4 antimicrobials for a long-term treatment, up to 6 months, leading to poor therapy adherence and antibiotics resistance. A vaccine exists, but it does not provide a complete protection. A new treatment strategy is needed to complete the goal set by the World Health Organization, ending the global tuberculosis epidemic by 2035. In this context, our laboratory has started to investigate potential combination of drugs already on the market to inhibit mycobacterial growth. The susceptibility of Mycobacterium tuberculosis to glycopeptide antibiotics, such as vancomycin, was recently discovered by our team when combined to other drugs able to target mycobacterial cell wall lipids. Tetrahydrolipstatin, also called Orlista, was the most interesting lipid-lowering drug. Indeed, vancomycin alone could inhibit 99% of mycobacteria at a dose of 100 micrograms per ml, while tetrahydrolipstatin alone had an inhibitory action at a dose of 25 micrograms per ml. Combined, the minimal inhibitory concentration, the MIC, of vancomycin was 10 micrograms per ml and 1 microgram per ml for tetrahydrolipstatin. This synergistic action of the drug combination was proved at a 10:1 ratio. One common characteristic of these two drugs is that they are poorly bioavailable. Therefore, combination must be administered directly in the lung to treat tuberculosis. Inhalation of this combination could be a good alternative. Local administration decreases systemic side effects and increases the drug concentration at the site of action. To date, inhaled vancomycin has proven its safety in humans, but no safety data has been published on inhaled tetrahydrolipstatin. The aim of the part of this project is to demonstrate the good lung tolerance of the potential anti-tuberculosis combination vancomycin-tetrahydrolipstatin at high concentrations under in vitro and in vivo conditions. The first step of this work was to know if vancomycin alone, tetrahydrolipstatin alone, or the combination had any impact on the cell proliferation at doses up to 125 times the combination MIC. The assay used was a MTT colorimetric test. As the aim is to administrate by inhalation, bronchial caliphree and alveolar A549 cells were selected for this assay, and the monocytes THP1 were also selected as a precursor of macrophages. On the left graph, results show that cell proliferation was not affected by the presence of vancomycin, even at the highest dose tested. However, tetrahydrolipstatin, graph in the middle, and the combination on the right presented a dose-dependent effect. The IC50 was reached for tetrahydrolipstatin and the combination at concentration 30 to 55 times the combination MIC. It is not possible to explain if this tetrahydrolipstatin effect on cell is permanent because cytotoxic, or if it's just temporary because cytostatic. To have a better idea of the type of tetrahydrolipstatin effect, a lactate dehydrogenase assay was managed. The aim was to determine the percentage of cytotoxicity of vancomycin, tetrahydrolipstatin, the combination on cali-free cells at doses up to 200 times the combination make. The cytotoxicity rate of all the treated group cells was around the baseline of the negative control at 0%. As tetrahydrolipstatin did not present any cytotoxicity in this assay, it was concluded that the effect observed in the MTT assay was caused by a cytostatic effect rather than a cytotoxic effect. After that, an in vivo experiment was then performed to evaluate the local tolerance of a subacute exposure to each drug and their combination. 3 groups of 16 mice were administered either 50 mg per kilogram for tetrahydrolipstatin treated group, 500 mg per kilogram for vancomycin treated group, or 50 mg per kilogram of tetrahydrolipstatin and 500 mg per kilogram of vancomycin for the combination treated group. They are treated 3 times a week for 3 weeks. These 3 groups were compared to the negative control group and the LPS positive control group. Isoflurane anesthesia in oxygen was administered to allow a direct endotracheal administration using a microsprayer connected to a high-pressure syringe. After the last administration, the trachea was cannulated. The bronchial alveolar liquid fluid was collected to conduct evaluation of local inflammation by ELISA assays. Lungs were also collected to measure tissue damage. The histopathology was conducted by an independent pathologist. During the study, no significant body weight loss was observed in the treated groups versus the negative control group, and no mouse reached the limit points defined by the scoring. Quantification of the pro-inflammatory biomarkers such as IL-1 beta, IL-6 and TNF-alpha was evaluated in BALF. As expected, the LPS positive control group showed a significant increase in these biomarkers. Within the treated groups, no significant increase was observed versus the negative control group for all biomarkers. Moreover, no major lung damage was observed by pathologic examination for the treated groups versus the negative control group. However, intra-alveolar hemorrhage was reported for all groups. As it also occurred in the negative control group, this damage could have been caused by the blood collection technique and therefore not related to real tissue damage. Other phenomena were observed only in the LPS positive control group, such as pneumocyte hyperplasia displayed for 50% of mice, tissue inflammation for 12.5% of mice, and bronchiolar epithelial vacuolation for 37.5% of mice. Therefore, vancomycin, tetrahydrolipstatin, and the combination did not induce any inflammation or lung damage and were well tolerated in the mouse model. The results obtained during this work confirmed previous studies on the safety of vancomycin delivered by inhalation. No cytotoxic effect of THL on cells at high doses in vitro was established with a potential cytostatic effect. This study demonstrated for the first time that tetrahydrolipstatin associated with vancomycin at a 10:1 ratio was not toxic on pulmonary cells in vitro or in vivo in mice during a subacute exposition. In conclusion, the combination is well tolerated and can be considered for inhalation drug delivery. Future studies will assess the efficacy of formulated combinations on a murine TB model. I thank you for your attention.